Hello. Good afternoon, ma'am.
Okay, ma'am.
Yes, good afternoon, ma'am. Okay,
Yes,
no
ma'am.
problem.
Nanti Bu, malah ini.
Yes, ma'am. Good afternoon, respected teachers and my dear fellow friends. On behalf of the Department of Medical Laboratory Sciences, I would welcome you all for being part of such an informative session, which is going to help us all understand an important skill called PCR technique. I am sure we all are well aware of what PCR technique actually is. This session aims to incorporate within us a better understanding of what working principle of PCR is, then a proper hands-on training with the thermocycler, understanding the potential applications of PCR in diagnostics, and as we all know that PCR has a great use in the modern diagnostic laboratory, this makes the skill an important requirement in the skill sets of laboratory and a research technician and an expert. I would like to thank our teachers, Dr. Monica Gulati ma'am, Executive Dean, lovely Faculty of Applied Medical Sciences, and the Head of Department, Mr. Gurinder Singh sir, Assistant Professor, Department of Medical Laboratory Sciences. Also, our today's speaker, Dr. Rohit Rai, Assistant Professor at Lovely Professional University, who has been awarded with prestigious awards, DST BRICS, Young Scientist Award, and DST IU, SSTF BA Award. And Mr. Dhruv Agarwal, Sir, Assistant Professor, Department of Medical Laboratory Sciences at Lovely Professional University. has gathered oceans and wells of knowledge within himself, and I personally admire Dhruv sir a lot. We all are blessed to have him as a teacher. Thank you so much, sir, for taking out time from your busy schedule for the benefits of your students. Thank you, sir. And over to you.
So, unmuted. Okay, okay. वो कहाँ जाना है? कहाँ पे? ये ये okay. It's a very good afternoon to all of you. Am I audible?
Yes, sir.
Okay.
Yes, sir.
Okay.
Yes, sir.
Good afternoon and welcome to all of you for this informative and interactive session on PCR. So PCR, I hope all of you are aware about the PCR, and somewhere you might have studied about the PCR also in your current classes as well, right? So can anybody tell me what is PCR? What is the full form of PCR?
Polymerase chain reaction, sir.
Very good. So PCR basically means polymerase chain reaction, right? So here on the slide you can see basically it is a diagrammatic representation, so which shows something. What it actually shows, I will tell you in a couple of minutes. So here you can see that there are two lines. These are getting multiplied multiple number of times, right? So this particular technique deals with multiplying your DNA. So these two lines basically represent the two strands of DNA, right? Now, when I say multiplication, so it means that making the copies of the DNA. So making multiple copies of DNA is very important, and it is required for multiple different things. As simple as that. For example, say you have your original certificates with you, right? And someday you go for some admission or for some job, and they ask you to submit, say, let us say 10 copies of your certificates. So what you will do? You do 10 Xerox copies, and how those 10 Xerox copies you will get? Some Xerox machine is there, a photocopier is there, right? So similar thing is over here. So this is a single DNA molecule. You can say you have, it is your certificate, right? And you want the multiple copies of that. For that you require one machine, a photocopier. So similarly, we have a photocopier here also, which photocopies multiple times your DNA, and that is referred to as the PCR machine or the thermocycler. Okay. So how it works and what are the basic principles and what are the various steps involved over here, we are going to focus over that, right? So this is a very basic kind of seminar, you can say, where we will be discussing about how a basic PCR machine works in theoretical concepts, right? And then we will try to implement those theoretical concepts into the practical aspects also through the hands-on practice session. Okay. So here you can see that there is one DNA molecule. There are two and this, and you are well aware about that. Two strands of DNA are there. One is a forward strand and one is a reverse strand, right? So both the strands are held up together through the various hydrogen bonds, right? So this is the which we will process our one molecule of the DNA, and ultimately you will get the multiple copies of the DNA. But before we go into the details of this, can anybody tell me who discovered this great machine or the process? Who is known to be the discoverer of the PCR?
Carry Muller.
Very good. So in nearly 1985, Carey Mullis invented this process or discovered this process, which was known as the polymerase chain reaction, right? So there is why we call it a chain reaction, because just like a nuclear chain reaction, the chain reaction goes on and multiple copies are being synthesized. Okay. And these copies which are synthesized, they are identical to your original DNA molecule. उससे भी होगी, मुझे लगता है नेटवर्क की यूट्यूब थोड़ा लैग पीछे स्लो चलेगा, हाँ थोड़ा पीछे चलेगा। वो यूट्यूब वाला थोड़ा पीछे चलेगा। गूगल मीट अभी चेंज हो रहा है,
हो गया अब हो गया।
मुझे नहीं लगता ये होगा। फिर ये हटा देते, स्लाइड शेयर हटा देते, ऐसे चलेंगे। अभी हो okay, so let's say there is time. It takes
Excuse me, sir, your voice is breaking.
Yes, sir, voice is breaking, sir.
Sir, your voice is breaking.
Connection was. Is it fine now? Am I audible?
Yes, sir, you are audible.
Yes, sir.
Okay. So let us say that there is a crime scene, and at that crime scene there is some sample, biological sample that has been collected by the forensic team. That may be in the form of sample, hair sample, or nail samples, right? So now they want to understand, or they want to identify that DNA, or they want to identify the criminal on the basis of the DNA identification, right? But the sample which they have, that contains a very small amount of the DNA, and for the identification purpose, they require a huge amount of the DNA, right? Some bulk amount of the DNA. So how they are going to get that bulk amount of the DNA? This is number one question. Second question is that after they have extracted the DNA, they want.
Sir, again, your voice is breaking.
Part
Again breaking.
okay. Voice break. Just wait. Okay, I hope it is clear now.
Yes.
Yes, sir.
Okay. So there are two scenarios. Number one scenario I explained you. Second scenario is, let us say that the scientists have extracted the DNA from any organism, let us say from fungi or bacteria, and now they are interested in studying about one specific gene. Let us say they are interested in studying the properties of or the expression profiling of insulin gene, right? So what they are going to do for that, or how they can take out that particular fragment of the gene out of the whole DNA, because DNA is a very big molecule, and taking out just a small piece of DNA requires some skills. How they are going to do that? So how the scientists will solve these two problems, that we will try to see over here. So for the solution for both of these is to amplify a small part of the DNA. For the forensic part also, the whole DNA they cannot handle, right? So we have in the different organisms genome certain specific markers are there, which can be taken as their specific identification feature. So those particular markers or those particular gene fragments can be targeted for the identification purpose, right? Let us say that if any organism specifically produces the insulin genome, let us say I am the only person who expresses the insulin. So just design the primers for insulin and try to put into the different DNA samples which you have. So whatever DNA sample will show you the presence of insulin gene, it means that that sample belongs to me. It is just like that. Okay. So now for amplifying a particular part of the DNA, you may proceed with the two ways. Either you can go for the polymerase chain reaction or you can go for the cloning. When you need the bulk amount or the multiple copies of a specific gene fragment or the DNA fragment, right? So cloning is a tedious job. It is not an easy task. It is a tedious job where what you need to do is you will take the whole DNA, the larger fragment of the DNA, and you will cut it with some molecular scissors that have some restriction endonuclease enzymes, right? And then you will take a specific fragment and you will ligate it with some vector, that may be plasmid or some other vector, and then you will transform that particular recombinant DNA into the host, right? And in those hosts, when those hosts will multiply, then your gene copies will also be formed, multiple copies will be formed. But this is not an easy job. It is a lengthy process and it is quite a complex process to be followed. On the other hand, we have a much simpler mechanism, that is the polymerase chain reaction, right? So what we do in this case is we proceed with the three basic steps and you get multiple, you can say millions of copies of DNA within 3 to 4 hours. Millions of copies of the DNA you will get within 3 to 4 hours. Isn't that beautiful? Rather than spending so many days and failing multiple times, you can simply amplify or make multiple copies in within no time, right? So that particular technique is a polymerase chain reaction technique, which is quite simpler method as compared to your cloning method, right? So in the polymerase chain reaction, there is nothing new which you don't already know. How many of you are well aware of the DNA replication process? DNA replication process. I hope most of you know about the DNA replication. Nowadays, you study these processes in 10+2, 10+1 also.
Yes.
Yes, sir.
So DNA replication process is an autocatalytic process of the DNA which goes on within the cell, where it makes its own multiple copies at the time. You can say it makes its own copy while the cell is going to divide, right? The same thing which when we setup, so that is the name given to polymerase chain reaction. So whatever techniques we have designed scientifically or in the artificial manner, so all are inspired by the natural processes themselves, right? So we will try to correlate over here. So this is a DNA replication process. A brief summary of that I will give you. So what happens over here? The double-stranded DNA is first of all opened up. These are two strands of the DNA. First of all, they unwind, right? That is what happens. It unwinds. There is a group of enzymes which are involved in that for opening up this double-stranded molecule. Then what happens? One RNA primer goes and binds to a specific site over the one of the strands. This RNA primer is important. Why it is important? Because it provides a 3' OH group, free 3' OH group, which is required for the activity of DNA polymerase enzyme. Now this DNA polymerase enzyme will come and bind at the 3' free 3' OH end of the primer, and it will start synthesis of the strand from 5' to 3' direction, right? And meanwhile, what it does, it collects the nucleotides from the surrounding environment and keeps on putting them. And these nucleotides are added in a complementary basis. Complementary means if A is there, so T will go over there. If G is there, so C will go over there. So complementary base pairing is there. Okay. So the same thing we have tried to, or the scientists have tried to replicate in the artificial setup. And the same thing is followed in the PCR reaction. How it is done? Let us have a look at that. So here the three important questions are there which you must understand. And the solution for these you must look for in this webinar, right? The first is how the amplification will be done of a specific gene. Secondly, how you will determine your target sequence, that which is the amplified. Then how the amplification will be specific for the certain segment. Okay, fine. I have done the PCR, everything is done, but how can I make sure that the product which I have is that specific gene only for which I was looking? That is also important. Maybe you were looking for insulin and you ended up with something else. Then the whole process and the whole purpose is lost, right? So for the amplification of a specific target sequence, what you need is, first of all, have you ever gone for the photocopy? So what you get in the Xerox copy, whatever you have given. So whatever you have given is actually a template that you want multiple copies of that template. So that specific DNA sequence you must know, and that specific DNA you must have with you from which you want to amplify a specific gene. And that specific sequence is referred to as a template DNA, right? And this template DNA is targeted by the PCR primers. The PCR primers are basically artificially synthesized small, you can say that nucleotide sequences which are used to initiate the synthesis of artificial DNA. So here basically we are artificially synthesizing the DNA, multiple copies, right? So it does require the knowledge of some DNA sequence information which flanks the fragment of DNA to be amplified. Now, what does it mean? It means that, for example, say if this red colored box indicates or shows you the gene which you want to amplify. So apart from the sequence, you must have some knowledge about the surrounding DNA sequence also. Then only you will be able to design the efficient primers, right? So the primers are going to bind at this flanking region, and your polymerase will start the synthesis from here in this direction, and from here in this direction. So the knowledge of the flanking regions of your target gene or gene of interest, I would rather use the term gene of interest, it is very important. For the synthesis, what you need to do is you need two primers. A pair of primers is required, and these two primers, they are going to bind at the 3' end of the DNA strand, template DNA strand. It is not going to bind over the 5', but it is going to bind on the 3' strand of the DNA. Okay, on both the DNA. Now the question arises over here is that why do we need two different primers? Why we cannot work with a single primer? So here basically, first of all, you must understand that the two primers which we use are the forward primer and the reverse primer. So what does forward and reverse primer means? Forward primer means that the sequence of this forward primer is similar to the forward strand of the DNA. Say this one moving from 5' to 3' is the forward strand of DNA, and 3' to 5' direction, it is a reverse strand of the DNA. Out of the two strands, one is forward strand, one is reverse strand, right? So the primer which is having a sequence similar to the forward strand of the DNA, template DNA, is referred to as the forward primer. But the interesting point over here is that this forward primer will not bind to the forward template strand, because sequence is same, not the complementary. So its complementary sequence will be present on the reverse strand. So that's why it is going to bind the reverse strand. Forward primer going to bind the reverse strand, and reverse primer will bind the forward strand, because their complementary sequences are present on the alternate strands. Any doubts on this one? You can see, let us say that this was the sequence at the 3' end of the forward strand, that is PAA. your reverse primer will have ATP, right? So that's why it is complementary over here. That's why it is binding here. But if you see this sequence of the reverse primer is actually similar to the reverse strand, right? So that is why it is referred as the reverse strand, but it is binding to the forward strand because of the sequence complementarity. So the same thing goes for the other one also. I will tell you. In the DNA replication process, I told you that one RNA primer goes, it provides the free 3' OH end from where the DNA polymerase will bind and start the synthesis of your DNA strand. So you can say that free 3' OH is the starting point of the DNA polymerase. And this primer is providing that starting point to the DNA polymerase. Okay. Now the question is, if we have only one primer, so in the reaction during PCR, you will be able to get just one strand synthesized. But we need both the strands to get a complete gene sequence, right? Or intact gene sequence. We need both the strands, but if you are using only single primer, you will end up with the single strand synthesized. Okay. So that's why we need the two primers, forward and the reverse primer. Here you can see the different components which are required for the PCR reaction. So one is a DNA sample, that is a template DNA which you need, from which you are going to copy the sequence for the gene. Then you require the primers, which will act as a start point for the DNA polymerase. Then you require the nucleotides. So these nucleotides we provide artificially as the DNTPs, dinucleotide, deoxynucleotide triphosphates, right? So all these are provided artificially in the reaction by us, right? Then you need the DNA polymerase inside. So Taq polymerase is one of the DNA polymerases, or you can say it is the most ancient DNA polymerase which was invented first or discovered first. Why it is referred to as Taq? Because it was isolated from a bacteria known as Thermus aquaticus. So T for thermus and AQ for aquaticus. Okay, so that's why it is referred to as Taq polymerase. Then this is the mixed buffer. This is a mixed buffer. This actually provides you the balanced pH and electrolyte solution, an environment which is required for the proper functioning and stability of the DNA. Okay. Then this is a PCR tube in which we make up the reactions. We will make you practice this thing, how to make the reactions also, right? And apart from all this, magnesium chloride is required. Why magnesium chloride is required? Magnesium or manganese chloride are generally added. Why we add those? Yes.
Is a bivalent and they act as gelatin agents.
Okay.
Co-factor for the activity of polymerase.
So this Taq polymerase is an enzyme, right? So do you guys understand the concept that enzymes, they sometimes require some cofactors for the proper functioning, without which they may not work at their actual efficiency, right? So these magnesium chloride or manganese chloride factors for this enzyme, DNA polymerase. That's why they are required in the reaction. Then DMSO, that is dimethyl sulfoxide, is required to avoid the formation of the secondary structures. Secondary structures means you are adding the primers. So maybe you get the primer dimers over there. So the primers in the reverse order, they may also sometimes form the pairs, right? Or the DNA molecules, they may ligate back. So to avoid those type of the secondary structure formation, inhibit basically the additional structure formation. Otherwise, when you are done with this, you have multiple things in your reaction mixture apart from the amplified DNA only, right? Taq polymerase is the DNA polymerase enzyme which is going to synthesize the strand. Just like DNA polymerases in your natural replication process synthesize the strand, so this Taq polymerase is playing role over here. Then this is the PCR cycle. So the PCR actually takes place in 3 different steps. And these 3 different steps are actually differentiated on the basis of the temperature. The first step over here is the denaturation. Now just try to recall what I told you about DNA replication, and try to what we call it. Try to what you call it. That particular word is not coming in my mouth. Correlate. Yes, just try to correlate it with the DNA replication. Right. So first step is denaturation. So in that denaturation, what we are doing is we are giving the high temperature treatment. So this high temperature treatment, what actually it is doing is it is helping us in separating out the 2 strands of DNA. The same thing was done by some helicase enzyme in the DNA replication. Here we are not using any enzyme for that, right? Because those enzymes may not withstand such high temperatures, or they may not work efficiently over here. There are different ways of doing the denaturation. Alkali can also be used, right? But they sometimes chemically modify the structure of DNA, so we are not using that one. So what happens over here is, in the denaturation, the 2 strands they separate out. They are made up of, they are linked together by the hydrogen bonds. So those hydrogen bonds are melt away, they break down during this denaturation process, and 2 strands they separate. Then the second step is annealing. If you remember, I told you the DNA application that one primer will go and bind. So same thing happens over here also. The primers which you have designed, you have added in the reaction mixture, they will go and bind to the flanking regions around the 3' end on both the sides of the gene, right? Both the strands of the DNA basically. So this is the annealing step where your primer is going to bind with the DNA strand. So you can say a sort of hybridization is going on. DNA hybridization. Are you guys aware about hybridization? DNA hybridization, RNA hybridization. Is there DNA RNA hybridization also? DNA RNA hybridization also takes place? Yes, anyone? Have you ever heard about DNA RNA hybridization? No?
No, sir.
But what if I tell you that DNA RNA hybridization also takes place? It takes place within your body also. Are you aware about the process of transcription? Huh? So RNA strand is synthesized complementary to DNA strand. At some point of time, that DNA is attached to the RNA. That is DNA RNA hybridization. Isn't it so? It happens, right? It happens. Then there is the third step, that is extension. So what happens at annealing? Your DNA is well settled, right? The whole template is ready. Primer has attached itself. Hybridization has been done. Then your DNA polymerase now is going to bind over there, and it will extend the strand. Artificial synthesis of the strand will take place. You can see different temperatures are there. The first step is at 95 degrees Celsius. Annealing is at 50 to 65 degrees Celsius. Actually, this particular temperature depends upon a couple of things. The primers which you have designed, TM of the primers, it will depend upon that. And TM depends upon what? Yes, TM depends upon what?
GC content.
GC content, very good. So GC content, higher the GC content, higher will be the melting temperature. Why so?
Because of triple bond.
This is ATGC. So G and C content, if it is high, so high temperature you require, high energy you require, because G and C, they are linked together by 3 hydrogen bonds. A and T are linked together by 2 hydrogen bonds. So obviously you require greater energy to break 3 hydrogen bonds as compared to 2 hydrogen bonds, right? So that's why if greater the GC content, greater will be the melting temperature. And annealing temperature has to be lesser than your melting temperature. Because if you are keeping the melting temperature and annealing temperature same, so your RNA will never be, your primer will never be able to bind with the DNA, target DNA, right? And then you will say that the primers are wrong. It is not any fault with the primers, but it is the fault with the selection of right temperature, which is there. Then extension we have done at 72 degrees Celsius. Why we do the extension at 72 degrees Celsius? Yes.
Because of optimum temperature for the enzyme.
Perfect. I hope you guys understand that all the enzymes, they have a specific temperature for the optimum activity. So this 72 has been selected because it is the optimum temperature for the activity of DNA polymerase, the tech polymerase, right? So that's why this temperature has been selected. Your enzyme may not work. There may be misfolding, or you can say the coagulation of the protein. So what happens? Enzyme is basically a protein only, na? So in the sterilization, you may have studied this thing. So heat sterilization is there. What does it do? It coagulates your protein or so same thing happens over there. Enzyme activity is lost basically. So you can see over here clearly that the hydrogen bonds have broken. Hydrogen bonds have broken, and the two strands have separated at the high temperature in the first step, that is denaturation. Right. Then in the second step, you can see that the primers, they have bound. The primers, they bind to your DNA template strands, both the strands. And in the third step, this particular DNA polymerase comes and starts the synthesis of the DNA over there. How you will confirm that? See, we have taken the temperature in such a manner only which is sure shot that will separate out here. Around 90 degree or somewhere, your two stand, the hydrogen bonds, they melt away. The energy required is lesser, but we are cautiously taking the more temperature. That more energy is there, and it sure shortly it dissociates the bonds over there. Okay, so everything is thermodynamically studied first. Then here you can see that the DNA polymerase is further synthesizing the strands over there. So DNTPs are providing the bases to be added over there, and DNA polymerase is continuously adding those bases. Right. So this is a program, how we fix a program in the thermocycler. So in the practice, probably Dhruv sir will tell you how the program he has set. So you can see over here. So denaturation step is there, then primer annealing step is there, then extension step is there. If you see denaturation and denaturation is the overall denaturation of the DNA initially we have done. Then in the each cycle, so there are multiple cycles in which we carry out this. So the first cycle will start from the step number 2, which is the denaturation at 94 to 96 degrees Celsius for 30 seconds to 1 minute. Okay, so this much time is required to dissociate the two strands from each other. Then primer annealing, the third step, 50 to 70 degrees Celsius, and this is for 30 seconds to 1 minute, which is sufficient time to allow the primers to go and bind with the DNA strands. Then extension is there at 72 degrees Celsius. Now one thing over here is that how the DNA synthesis stops, because DNA polymerase is also there, nucleotides are also there, all the favorable conditions are there. So the enzyme will continuously keep on synthesizing the strand. But how does that reaction stop? That basically depends upon the time which we have given for the extension. If you know that the size of your gene is what, if you exactly know the size of your gene of interest which you are going to copy, you can decide what time you should give for the extension step. Generally, we give 1 minute for 1 kb. 1 minute for 1000 base pairs we give. If your gene is 1000 base pair long, you will give 1 minute for that. If it is 2000 base pairs long, you will give 2 minutes for that. But in any case, we keep it minimum to 45 seconds. Right? 45 seconds minimum we give it. So these are the 3 main steps. After this, what happens? Step 5 is the repetition. So we give a loop over here. We fix the loop in the thermocycler so that step number 2, 3, and 4 they are repeated multiple times, and we actually write over here that how many times we want. Let us say we want to repeat it 30 times. So we will fix the time as cycle as 30. Every step will multiply your DNA. Na? So how many times you want to multiply your DNA? Lesser the amount of DNA initially you have, more number of cycles you will need to give. Okay, you will need more amount. Huh? So lesser the initial amount you have, more the final the cycles you will have to give. Let us say that you have the 30, you have given the 30 cycles. So 30 times step number 2, 3, and 4 will be repeated continuously. Once all these 30 times these 3 steps have been repeated, then there will be a final extension. Final extension again at 72 degrees. They are properly fixed over there. They are stable now. So this completes your PCR process. After that, we have a holding. Holding you can do either at 16 degrees Celsius or 4 degrees Celsius. 4 degrees Celsius when you have to store for a long period of time. Like for example, say if you have set up the PCR reaction in the night and you are going to look back in the morning, so you can keep it at the 4 degrees Celsius so that there is right. Otherwise, if you want to hold it for 1 to 3 hours, 16 degrees Celsius temperature is enough for that. Okay. So this is the whole PCR process. Then how we carry out it. You can see over here. So a double-stranded DNA molecule, denaturation step at around 90 degrees Celsius or 94 degrees Celsius, two separate stranded. Here around 50 degrees Celsius, the primer, the extension of the primers began. So this is one cycle. So multiple cycles we give. From in first cycle, you got 2 copies from one DNA, then further 2 more copies over there. Like this, it is going on. Now, one question arises over here that how you will get to know that at the end of your PCR process, how many copies of DNA you have. How you will get to know that? Is 2 raised to the power n. 2 raised to the power n, right? So I will not be able to write over here, but it is 2 raised to the power n, where 2 means at every step your DNA is making 2 copies. So 2 at the base, and n is actually the number of cycles which you have given. Okay. Now can you guys do a quick calculation for me? Let us how it will be 60. 2 raised to the power. Can you guys quickly do a calculation for me? Let us say that initially you had one DNA molecule only, right? And let us say we have given it 20 cycles. So what will be the number of DNA molecules which you will have at the end? Yes. How many copies you will have at the end? If initially you had 1 copy, do a quick calculation. What formula you are fitting? Just tell me the formula first. How much? 1048576 576.
Yes, sir.
Okay. So is this the answer which everyone got?
Yes, sir.
Okay, I'm not having calculator right now with me. So what you have to do is you have to do 2 raised to the power 20. Calculate this, and you are done. Okay. So this is how many copies you formed. How many copies are formed, and how many total DNA molecules you have right now. You have to add 1 into that also, because the original one is also there, na? Or dissociated. That is fine. So these are the original number of copies which you will have at the end. Okay. Okay. So this is these are the important steps in performing the PCR. This is quite. I cannot even read out from the distance. So identification of the location of the target sequence in DNA template. So what particular sequence you have to amplify, right? So that you should identify first. And that could be, let us say, any gene of your interest, right? Then there is primer designing. So we will have a separate workshop sometime where we can tell you how to design the primers, because it is a little bit of a bioinformatic thing. So we can have a separate session for that sometime. Then we have the PCR optimization. That what should be the different PCR conditions, temperature, and time for the different steps. That has to be optimized. Then this is, can somebody quickly read? Then post PCR result analysis, right? Because there was another question that how you are going to figure out that your particular gene of interest has been amplified. Number one, whether any gene has amplified or not, and secondly, if the gene has amplified, is it exactly your gene which you are looking for? These two things we have to figure out, right? Then there is PCR troubleshooting. If you are not, maybe everything was fine, but you did some mistake. So that is a troubleshooting which you have to do. And then this is start your PCR and what is this? And visualize the results by AGE. What is AGE?
Agarose gel electrophoresis.
Agarose gel electrophoresis. Good. Right. Agarose gel electrophoresis. So after completion of the PCR, how you will make sure that your target know your product size. You must know. Right. You must know. For example, say if your target sequence is 350 base pair long, you know if it is 350 base pair long. After PCR, you run the agarose gel along with the marker, and you can see over here the gene product over here is again 350 base pair long. So this confirms two things, and number two, it is exactly the same gene which you are looking for. Okay, so that's all I have for the session, and if you guys have any queries, you can ask me, or otherwise we can switch to the practical sessions. So, Rahul? Definitely.
No, sir.
Okay, guys, then now we will have a short break of 10 minutes, and you can go for, you can take it as a bio break. So after the short bio break, we will start the practical session by 3:10. Okay.
Okay, sir.
Okay, sir.
Bye, sir.
Thank you, sir.
ये दिख रहा है मेरा सेकेंडरी डिवाइस? क्या दिख रहा है? कहाँ रखा था? यहाँ रखा। अब दिखा मैं मशीन। ये बेस्ट है। So let's start the practical work. So tell me what do we require for PCR?
Sample.
One by one. And one person will give me the one component only. Blood sample. Okay. And
Sir, primers, nucleotides, and then just take to polymerase.
Polymerase, like polymerase, any polymerase is a acceptable answer. But there was something.
And also some mixed copper. Mixed copper.
Okay. And
Nucleotides को वैसे ही करना था.
DMSO.
PCR machine है.
That's it.
Samples.
There is no matter of laughing. So let's say gloves. Why do we need gloves?
Protect our skin from.
Any bacteria.
सर आपने कहाँ से कैसे करेंगे बच्चों की आवाज आवाज नहीं आएगी ना आपकी
हेडफोन पहन लो.
So this technique is very important, right? So please, it's a good opportunity that you got, that you are having opportunity to perform this, right? So whatever he is telling you, make sure that you understand each and everything. And if you don't understand anything, please ask him, right? It's a good platform. We are like a family. You can ask your stupid questions also. No such issue, right? Sometimes we ask to ask that question as if someone is going to insult us. No one is going to insult us. Okay. So whatever question is coming, just ask him. He is there to clear the doubts, right? So take this as a good opportunity and make sure that you clear all the doubts that come in your mind regarding this year. Because आपको है उसका subject है. Do you have this technique? I think there is no subject related to techniques. Next semester, for masters there is, and for BSc also maybe next semester there will be a subject where you are going to learn all the techniques, and there will be one technique PCR. Okay, and hopefully we are conducting next session on another technique, another molecular technique. Once it is ready or once it's final, I'll share it with you.
Is it okay now? So the answer was bacteria. We require gloves so that we can protect it from the bacteria.
No sir, this is ना जो हम sample लेंगे वो contaminate हो जाएगा.
Okay, contamination of sample, but we are providing it 94 degrees Celsius for 5 minutes. It will kill most of the contamination at that particular step. So what else?
ये सब तो निकालिए ना तो फिर सर जैसे हमें पता करना है इससे और
Okay, our DNA will be again protected because we do not DNA sample or the PCR amplified sample above 4 degree. We always incubate or place it at either -20 or 4 degree. And we know most of the metabolic activity are hindered at that particular temperature. So they are not acceptable answers. Try more.
something that we can see
Something is that, yeah, RNAs or DNAs. Our skin is rich in those particular enzymes. What they do, they degrade the bonding between two nucleotides. They chop off the nucleotides. So we are trying to create a chain which is made up of nucleotides, and we incubate some enzymes which is chopping off that chain. So our motive is completed or not? No. You know what? Yes, sir. It is a question. Either yes or no is the answer. If you are giving me a yes answer, then our motive is completed, so that you can chop off the nucleotide chain. Is that so? If, so right now in the PCR, our motive is we have to chop off the nucleotide chain. Okay? But in denaturation are the nucleotides. We will keep this question in mind because the previous concept was wrong actually. Our motive is not to chop off the nucleotides, is to amplify the nucleotides. Okay? What is our aim? To amplify the nucleotides or to amplify a particular strand. Okay, so what is being carried out in a polymerase reaction? We are adding nucleotides one after another so that we can create a chain of particular length. Okay, so keep this aim in mind. Okay, what was your question? Antibody denaturation. Okay, so at denaturation, those DNAs and RNAs are mostly stable, so they will not denature that easily. Or if we talk about the structure of RNAs, they are made up of RNA molecules itself. Okay, so they can fold and unfold. So along with the reactions of annealing, denaturation, and other, they will also be distorted. Again, coming to their original structure, carry out their function, and at each step they will chop off the structure. Okay, so what else we require? We require magnesium chloride.
Right.
And if we break down the magnesium chloride, which ions will form?
Magnesium
Magnesium
and chloride.
and if we talk about which particular metal ion is responsible for the enhancement of the activity of the
Magnesium.
magnesium, because it is a metallic ion enzyme. Okay, and if we talk about the condition, either in vitro or in vivo, this PCR reaction is an in vitro or in vivo condition.
In vitro.
What is an in vitro condition?
In
So we have to provide each and everything so that we can carry out our aim. We can carry out a particular thing that we are going to perform. Okay, so step by step, what we do, we require a particular substrate, which is our DNA template. DNA template. And the DNA template can be of large size, small size, any size, but we must be selecting a particular in between area that we are going to amplify. We have to target our primer at a particular site of that sequence, and let's say we don't know whether this particular molecule is complete or incomplete. We have a primer, a forward primer, which will bind to the one step or one portion of the DNA. And we have a second primer or third primer, which portion is missing in the molecule, whether this PCR reaction will occur or not. We have a incomplete stretch of substrate in which the site for forward primer is present and site for reverse primer is missing. So the reverse primer cannot bind because no complementary strand is there, and else only one particular strand is there which is already incomplete. Okay, only the half reaction will carry out. Can this condition will lead to some formation of molecules or not? Yes. Okay, how? Try to draw a mesh, yes. Yes, because half portion is there and they will be forming the molecules. At least one strand will be carrying out the proper functioning. Okay, But what has observed, sometimes it do not give any product. So why is that? Because the both strength, or if you talk about the reaction, the exponential reaction versus linear reaction. So if you have only one strand which will be amplified, that reaction will go or will run only in a linear fashion. 1, 2, 3, 4, and 5. Because after each cycle, your second strand will go into dustbin or waste. Only after each cycle, only one molecule will produce. So either you can run your PCR cycle for lakhs in lakhs cycle, then only you can get a result. But this thermocycler will burst and for those number of cycles. So usually if you have only a specific one molecule which will be amplified, not a two molecule, you will not get any results. Why? Because that particular reaction will be only linear, not the exponential. Okay. So and we require primers that will bind to the specific on the substrate.
It's a forward primer.
Next, what we require?
Sir, both forward primer and backward primer.
Primers and
Yes.
okay, so if I take the template and forward primer and reverse primer, will my polymerization reaction work?
Yeah.
That polymerase and
DNA.
so what do we do? What do we try to create in an in vitro condition? So can we design any in vitro condition just as such, so without any prior information? We have to mimic the situations of in vivo. So in vivo condition, what is being used? Different and complex machinery is being used. We have to design. So I'm just telling you how this small molecules and how those 6 or 7 specific molecules we select only. Why not the other molecules? We can have types of temp polymerases, type of other molecules also. That take example of DMSO. We can have different type of chemicals also for the same property, but are they effective or not? So in the practical work, what we select that particular chemical or that particular substrate which will give us the broad range results. Okay, so this DMSO is effective in most of the cases. Okay, so if someone is asking, can only DMSO be used? No, if that specific question is in front of you, we can use other chemicals also. So it is one of the chemicals which can be used. And why we use DMSO? So if you have a particular strand which is single stranded of that particular single strand molecule, molecular biology is nothing just the imagination because we cannot see anything. Why? So this is the DNA. So it will look like water and you can say it is a water, but let me assure you it is not water. Okay. So it is a DNA. Can you see this DNA? No. And we have here this tag polymerase enzyme. Can you see any tag polymerase enzyme? We have buffer. So it is already the maximum. The 95% of this buffer is already water. Okay. I will show you the original kit also. We can have different kits from different manufacturers. Like the kit we have in the laboratory is manufactured by company High Media. High Media. So you can have different kits according to your need. There are different sizes available for 50 reactions, 25 reactions, 70 reactions. According to that, whenever you are working or what is your aim for the reaction, you can order that particular kit. Okay. So this is buffer. And in buffer, what different components can be present? Like electrolytes or some osmoregulators. Okay. And we have here DNTPs. So what is this DNTP?
The nucleotide
Deoxyribose. D, the full form of D is complete deoxyribose. What stands for N?
nucleotide
Nucleotide can be.
and
So they are in dimer, trimer, monomer. Monomer. So either it can be GTP or ATP
Yes,
or
yes,
CTP
CTP.
or UTP. Are you sure UTP? So which reaction we are carrying out? PCR. And PCR is the mimicking of replication process. And in replication, we have T and V. Okay. Why do we require this DNTP solution? Add a single nucleotide one by one by the enzyme
That
flag
polymerase.
polymerase. Okay, so this will mimic the RNA polymerase tree. Okay, sorry, DNA polymerase tree. Okay, tag polymerase is one of the polymerases from the organism Thermus aquaticus. Okay, so there is no surety during the performance that the reaction or each and everything is perfect. Only the surety will be given by provided by you the results shown by gel electrophoresis. And even in the gel electrophoresis, if you perform gel electrophoresis without incorporating any compound which will give you color after binding with the DNA, you cannot see any results even in the gel electrophoresis. Let's say you are preparing gel and you forgot the compound that will give you the color during binding with the DNA. So you will not get any result and you will consider that my reaction was failure. But actually what happened? You forgot to add the compound which can be acridine orange or ATPR. Okay, so after binding with DNA, they will give you some fluorescence. Then only you can assure that the PCR was successful. Okay. Now that success rate will also depend on what is the size. So sometimes or many of the time, or let's say 99.99% we know the size of our amplicon. Amplicon is a term we give to the results of PCR. Okay, so 99.99% we know the size of that particular amplicon, what size we will get, and if we are getting that particular size, then only we are sure that our reaction was successful. Okay, and what else we require? We require MgCl2 primer and water. Okay, so here we have 10x buffer. Okay, so now here X represent a concentrated form. Sometimes in the biology we have X, which is telling us about the dilution, and sometimes we have X, which is telling us about the concentrated form. So here the X represent the concentrated form. Here the buffer is 10x. Okay, so if I design my reaction for 50 microliters. Okay, so for a 50 microliter reaction, how much buffer should I be adding? It is a 10 times concentrated buffer. 3 microliter. 10 microliter. So if I add 10 microliter buffer in a 50 microliter reaction, the final concentration of buffer will be 2x. And we have to make this concentration 1x, means unconcentrated buffer. So if we have the volume of reaction, the final volume of reaction is 50 microliter, and this is 10 times concentrated buffer. So how much buffer should be added? 5 microliter. Okay. And we have different concentrations of primer also. So whatever chemical we are using, we always have a particular concentration. Either it is a catalytic DNA. So what is the unit for the measurement of DNA? How we measure the DNA concentration? Microliter and in international unit. And so all of those answers are wrong right now. Any other? Molecular DNA. So you are half correct. You are half correct, don't half. You are half correct. If you try, you may get the right answer. What is mu? Yes. And you are also correct. We can convert microgram to kilogram to other gram. The basic unit of measurement will be gram, and that gram can be converted into different various ratios or different various other units. Okay, the basic that why we use the unit microgram, because the quantity always is very few, very small amount. So that is why we use the term microgram. Microgram per microliter, because it is something chemical structure which has some weight. And when if something have weight, we have to measure it by weight only. And that weight can be present in the form of solution at a particular proportion only. So we will say that this particular proportion is present in this particular proportion of the water or some other compound. Okay. So we always will tell us how many gram is present per how many ml. Okay. So we always denote this unit as microgram per ml, microgram per liter or microgram per ml. So it will always be the concentration how we want to denote that concentration. Okay. So usually which concentration is being utilized? It is being utilized in nanogram. Okay. So approx 200 to 400 nanogram per microliter can be added in the reaction, PCR reaction. So let's say you have 200 to 40, 200 to 400 nanogram per microliter template sample. This is your, let's say this is your template, not let's say this is your template actually. Okay. And the concentration is 200 to 400 nanogram per microliter. And you have to design a PCR reaction in which you have to add only 200 to 400 nanogram. So how many microliter you will use? Okay, if you are using 5 microliter, then you are adding 1 microgram to 2 microgram. And during the designing of PCR reaction, it's everything based on the how much compound we add. We know which compound we have to add, but how much? Okay. So approximately you have to add only 200 to 400 nanogram. And your stock solution is already 200 to 400 nanogram per microliter. So if you add by taking a reading on spectrophotometer, or we also usually give that particular estimate nanodrops. Yes, okay. A spectrophotometer will give you a reading at a particular wavelength. Okay. And for DNA molecule, we utilize a ratio of 260 to 280. The 260 is denoted for DNA or DNTPs molecules, or 280 is for protein molecules. So there is always a presence of protein along with the DNTPs. Why? Because they both are charged molecules, and if they are opposite charged, or let's say the more positively charged protein is present, so it will always be found in the close vicinity to that RNAs or DNAs. Okay. So now the question is how we determine the concentration. We always take ratio between 260 nanometer to 280 nanometer, and that ratio will give you a particular reading. Let's say 0.467. A spectrophotometer will give you reading like 0.467, 0.223, point something like that. Okay, now what you will do, you will purchase some standard, and the standard will be of known concentration. Let's say 1 nanogram, 2 nanogram, 3 nanogram, and those standard will give you a particular reading, and you will form a graph. 1 nanogram will give you this particular wavelength, 2 nanogram will give you this particular wavelength, 3 nanogram will give you this particular wavelength. Okay, you will form a graph between concentration and OD. Okay, now you can reciprocate how much OD will give me how much gram. Now you will be able to convert OD into gram. So you can know how many gram are present in my sample. Okay. So if per microliter mean in 1 microliter 200 to 400 nanograms are present, and if I have to add 400 or 200 nanogram, so I can just simply add 1 microliter. Okay. If I transfer 1 microliter from this sample to here, how many microgram will I be transferred? 1. That was the question. I am transferring the 1 microliter only. So if I am transferring 1 microliter, that is the 1 is being transferred. So in that 1 which is being transferred, how many gram is present? 200 to 400 nanogram. Okay. That was all about the capillary DNA concentration. Now the next is primer concentration. Okay, so whenever you will purchase any primer, they will give you a stock solution of microgram, and then you can convert the microgram solution into nanogram, and then the final concentration that you will use must be in the 200 picogram. Don't do it. Just purchase primer. Purchase primer. Dilute them 10 times and per 50 microliteration. That is why this workshop is being held. Okay, so there is another way: purchase some primer, dilute them according to the specification. There will always be specification how many microliter you have to add in that particular primer. Then 10 times diluted, and then directly use 1 microliter for 50 ml reaction. Am I correct or wrong? Wrong. Why? only very non-tangible mistake was there. Yes, 50 and later or something. Yes, we will try to find out. Yes, what should be there? Okay, nanometer. Can I add some liquid? Okay, can I add any liquid in picometer?
No.
The unit of liquid is liter. Okay, it is microliter, not 50 ml. Okay, the reaction volume is 50 microliter. It is on the screen also. So if you guys connected with the screen, so the composition. If you check the total reaction volume is 50 microliter. Okay. And in that 50 microliter, if your PCR reaction is very successful, so you by using 200 picogram of your primer and 200 to 400 nanogram of your DNA template, you can get 200 to 400 microgram or 10 to 100 microgram to 200 to 400 microgram DNA you can get for a 50 microliter reaction. Okay. So don't confuse yourself. Just remember the basic. Just remember only one thing: what we are doing and what we require for that thing. You don't have to worry about that.
No, sir. We will try.
Yes, sir.
What is the use of it? For DNA, we have 200 to 400 nanogram. Okay? And for primer, you have 200 picogram. So whenever you purchase a primer, the concentration will be in micromolar. Yes. You will always have to buy. You can form primer if you have very specific machine, so that you can add each and every nucleotide very specific. If you have, and if you purchase it, it will cost you approximately 17 to 20 rupees per nucleotide. Why a very good industry. And if you try to manufacture it by yourself, then I don't know what will be the calculation. You can calculate it according to your firm, and then try to input upon output, then efficiency you can calculate according to your firm. But if you order it, there's 70 to 20 rupees per nucleotide, you can get a very good primer. Okay, so it will always come in the micromolar, then you have to dilute it 10 times and then directly use 1 microliter for 50 microliter reaction. Okay, so right now what you can do, you can just forget about worrying this concentration. Okay, so you can directly use that information. Okay, so what you have to do for 10x, you can take 10 microliter of your primer plus add 90 microliter of your molecular biology grade water. Okay, so it will be your 10x. Here it is diluted. Okay, here the x represents dilution. Okay, and what else you require? You have template, now you have primer, and you have to have some enzyme. So here you have crack polymerase enzyme at 1 unit per reaction or 2.5 units per reaction of 50 micro. And what else you require? You require GNTPs. So you have 10 millimolar solution and you can add 2.5 microliter for 50 microliter reaction. And what else you require? This is polymerase enzyme. And here is your DNTP. And sulfur.
Master mix. Buffer.
And it is 10 times concentrated, so you have to add 5 microliter for 50 microliter. Okay, what else? MgCl2, how much is there? 2.5 microliter. And what is the concentration of that MgCl2? It is written after MgCl2 in brackets. $50 millimolar. Okay. And what else? Okay. So calculate, add all of the things. This 5 microliter, this 2.5 microliter, this 2.5 microliter.
DNSO. DNSO.
Already
Master mix.
this 10x offer 5 microliter, DNTP 5 microliter. Okay. So if we are adding 5 microliter for 50 microliter and the DNTP solution is 10 millimolar. So what is the final concentration of DNTP? How many times it is being diluted? It is 1 millimolar, and you can see the ratio, the unit is in milli, and here the final product is either in nanogram or in microgram. So you must be very sure about always have the high amount of your raw material, so it will not hinder the reaction. Okay. Add all of the things. 5 microliter acid buffer, 5 microliter polymerase, 1 microliter format primer, 1 microliter reverse primer, and 1 microliter of your template DNA. So what is the total? Either my math is wrong or yours. 18 microliters. Okay. And after calculating all of the components, so why after calculating all of the components? Because whenever you are performing any PCR reaction first time, so let's say you have designed a primer, you are carrying out the PCR reaction, and you will not get the result in first time. So you will have to optimize your PCR reaction by varying the concentration of DMTP or the primer. You can add 1 to 5 microliter, or the tag or this template DNA 1 to 5 microliter, or you can reduce the enzyme also. So after each and every optimization for a particular stretch of DNA, then you will get a particular amount of components that you will require, and then what is the final volume of your reaction? 50 microliter, and rest is 32 microliter. So for the preparation of 50 microliter reactor, you have to make a volume up to 50 microliter of the individual components. So you have to add 32 microliter of water for this particular setup. Okay. So any confusion up to now? Any question or confusion up to now? So if you are getting any confusion or reaction or any confusion in the mind about the reaction, so then let's forget each and everything. Just remember two things: what we have to do and what do we require for that purpose. We require these things. Then only you must ask the next question with yourself: how many amount we require those things, and then ask next question: how we can procure them, how we can purchase them, or how we can prepare them. Okay. If one second, please, for that particular setup. It will most of the time depend on the template DNA and your primer DNA. The most influencing components for a particular PCR reactions are template DNA and the primer setup. Okay. Any question, confusion? Okay, so let's prepare the reaction. So what I will be doing, I will be adding the components one by one in the reaction. So what we have, so might know, you might have known about all of these things. These are the PCR tubes. Okay, so these tubes are just which are very small in size. So roughly what do? We prepare the volume of PCR in very low quantity, either 25 microliter or 50 microliter. Okay, so and why we cannot produce the reaction in 1 ml or 2 ml, or why we cannot directly just design a reaction for 1 ml and it will solve our problem in a one go. So what most of the time what we do, if we are handling number of samples, let's say we have 90 samples, we don't have to waste 1 ml reagents. This tag polymerase alone for I think 100 units, you will have to pay 9,000 approximately, and this price is without GST. Okay, this is alone about this tag polymerase. So you have to purchase all of the components, and this 10 reaction kit is approximately 10,000. So 1,000 for one reaction is the estimated cost. So how much you are going to cost your patient? Let's say you have to diagnose a particular disease, you have a specific primer for that, and you have to perform that particular diagnosis in your laboratory. So are you going to charge 2.5 thousand for particular test? Will he pay you? Will you get more number of patients or more number of customers? Some of them may come and some of them may go. Okay. Let's say so this was the situation where you are preparing the reaction of 50 microliter only. Now you are thinking that I will prepare the 200 microliter reaction, so the cost will be goes 4 times. So it will be not economical. So what we can do, we can reduce the size
Hello.
of in gel for the running of gel for the detection.
Hello,
for a
good
test,
afternoon,
the
sir.
result will be either no or yes. If specific region is being amplified, means that defective gene is
Hello.
present and the person is being suffered from that particular disease. Okay, so ultimately what you require approximately 5 to 10
Hello.
microliter amplifier for a
Hello, Monica. Can
journey
you please mute
diagnosis.
yourself?
So you can use 25 microliter reaction also. Just divide each and every component by 2. Instead of 1 microliter primer, you can add zero point 0.5
Hello,
microliter primer. 1 microliter template, you can add zero point 0.5 microliter
Monica. Please
template.
mute yourself. Interrupting everyone.
And buffer will be added. How much buffer will be added for 25 microliter reaction? Yes. As you know, buffer come as a 10x, so we can add 2.5 microliter buffer. So it will give us the 1x concentration in the 25 microliter reaction. Okay. And what else? Okay, sometimes what we have to do, we have to take or we have to preserve that particular amplified region and we have to use it further. So we were discussing about why we do not do in a large quantity. Let's say we have to use that particular sequence or that particular amplicon for the further diagnosis or further, like let's say cloning. We require this particular amplicon in large quantities. Okay, but we cannot form or it is recommended that not more than 50 microliter reaction should be designed because in a thermocycler, first of all, in a thermocycler, only the half portion will be dipped. Only the half portion of the tube will be dipped, and you have to provide very specific temperature so that the very specific reactions can be carried out. Okay, and you cannot have very large amount of samples in the tube. Okay, so like I can demonstrate you on only one tube, and then you can individually prepare the reactions, and then I will tell you about the machine. Okay, so first I will prepare the single reaction, will tell you about the machine, and then you will be preparing the reactions, and then we will put your samples in the next cycle of the thermocycler. Okay, so is it clear up to now what we are going to do and how we are going to do? Okay, so let's take a short break for 5 minutes and then we will prepare the reaction. Okay. I have that.
Is the class going on?
ये हमारे आपके इक्वेशन में आपके राइट जानते हो डिफरेंस जब तुम डीएनए को घटाओगे तो नो yes you did the word answer did the word then करके उसको मतलब चेक करेंगे उसके डीएनए को अपनी लोग के डीएनए को चेक करेंगे हमें तो पता ही नहीं आज वहां से लीला होगा कुछ तो लीला होगा
No, sir, please zoom your video, sir. I'm not still clear. No, sir, you are not audible, sir. Hello, sir. Hello, sir. Sir, you are not audible. Hello, sir. Sir, you are not audible, sir. So reconnect
Thank you, ma'am.
Thank you, ma'am.